Hello everyone, welcome to the ITLS Academy. Today we are going to discuss another experiment, that is your detection of the adulterant in an edible oil. Today the experiment that we are going to perform is a detection of the mobile or a lube oil in an edible oil. Here a mobile oil are act as a adulterant and our edible oil is what our this is our sample. So we take a mustard oil as a sample and we detect that is there any mobile oil are present into it or not. Okay, so before starting this experiment we have to know that what does this adulteration means. Simply, the adulteration are defined as the addition of the chemical compound into a food product to increase its quantity, to increase its flavor, to enhance its color. That is called a adulteration. And when a adulteration occur, the quality of the product are decreases, and even sometimes the nutritional value of that product are also decreases. So here, now the second thing is that uh, during the adulteration process, the chemical compound which are added are known as a adulterant. Adulterant is always sometime a chemical compound, and sometime we interchange the good ingredients of the food product with the bad ingredients. Okay, if we talk about the black pepper. Okay, now in black pepper, whole black pepper, the papaya seeds are present. So what they do, they interchange the good ingredients and the bad ingredients of the food product. Okay, that is known as the adulterants. Now, for performing this experiment, we required this setup. And before introducing this setup, I have to tell you about why this mobile oil are added into the edible oil. Obviously, for a greasing purpose, for enhancement the quantity of the product, and for enhancement the color of that product. But this mobile oil have a adverse health effect. That's why it come under the category of the that's why it come under the category of the adult trend. So for performing this experiment, we required two chemical. One is your alcoholic potash and another one is your Gibbs reagent. This is your Gibbs reagent and this is your alcoholic potash. Now, first of all, we have to know that how we prepare this alcoholic potash. So for this alcoholic potash are also known as a alcoholic potassium hydroxide solution. Alcoholic potash are also known as a alcoholic potassium hydroxide solution. So for this we take a 70 gram of the potassium hydroxide and it will be dissolved into a 70 ml of the distilled water. See, the amount of the potassium hydroxide and the amount of the distilled water is always same. If you take 2 gram, then you have to take a 2 ml of that distilled water. And here the pellets of the potassium hydroxide we are taken. So when it is completely dissolved, then we add a 2 liter of the 2 liter of ethyl alcohol. Okay, then we kept it for a overnight and we filter it if it is required. Then we store it into a plastic bottle. So this is how we prepare this alcoholic potash. Now the second reagent is your Gibbs reagent, that is also known as a dichloroquinol chloramide solution. For the for this we required a chemical that is known as a 2,6-dichloroquinol chloride, 1 gram, and it will be dissolved into a 200 ml of the absolute ethyl alcohol, and then we kept it at 10 degrees Celsius, and this solution is only used within a 5 days. Then we required a burner, micropipette of 1000 microliter or 1 ml. Then we required our test tube stand, micropipette tip, and test tube holder. Okay, because when we are heated, then we have to use this holder. Now what we have to do, we take our 2 ml of the oil as a sample, into which we have to add a 1 ml of this alcoholic potash. Then we heat the tube into the burner. Okay, then it is decolorized. After that we add a 1 ml of this Gibbs reagent, that is dichloroquinone chromide solution. After that, we again warm the tube into the burner. Then if blue color are appear, then it indicates that the our sample are adulterated with that mobile oil. So let's start the sample. First of all, take a test tube. Then we have to take 2 ml of oil. We have to avoid the bubbles formation, okay? Because if the bubbles are formed, then error will occur. Then after that, we have to add the alcoholic potash into it. Take 1 ml alcoholic potash and add into it. Now after that, we have to heat it for a 2 minute in a burner. Then this color will be disappear. Okay. Now we will see the result after 2 minutes, because we have to heat it at least for a 2 minute. See that when we are heating it, its color are disappear. Okay, it is decolorized. Now after that, when its color are decolorized, then we have to add a 1 ml of Gibbs reagent into it. This is our Gibbs reagent. Okay, if this mobile oil are present, then it turn blue color. Here, now we have to warm it. Now see, there is no development of the blue color. If the blue color are developed, then it means that the mobile oil are present. So this is all about the detection of the mobile oil in a edible oil. I hope you will understand, but if you have any query related to this experiment, you are free to ask me in a comment box, or for a further detail, you can follow the ITLS Academy on different platforms. That is your Instagram, Facebook, Twitter, YouTube, WhatsApp, and LinkedIn, and subscribe our channel ITLS Academy on YouTube and get our app from a Google Play. You can also visit our website, that is www.itlsacademy.com. Thank you for watching. Please like, share and subscribe our channel.